Welcome everyone to the Demography Today lecture series, sponsored by the BBVA Foundation and organized in collaboration with the Spanish National Research Council, the LOMPOP Horizon 2020 project. And we are here today in Seville to listen to Helena Vezina. That it's a pleasure to have her here. And she will be talking about the i-BALSAC infrastructure. Helena Vezina holds a PhD in demography from the Université de Montréal, and is professor in the Department of Human and Social Science at the Université du Québec, Chicoutimi. Her research program touches on issues of historical demography and population genetics in multidisciplinary context. She has worked extensively on the genetic and ethnocultural diversity of Quebec regional populations and on the demohistorical factors that have shaped this diversity. She has also been involved in projects on the genetic epidemiology of various disorders. Her research rests on the use of genealogical and family data found in the i-BALSAC database. As you will listen with this, i-BALSAC database is a large database covering almost 400 years of history of the Quebec population, which she has been using since her doctoral research on the demographic and genealogical aspect of Alzheimer's disease. Since 2010, she has been director of the i-BALSAC project in charge of the development and exploitation of the population database, and she's currently working on a new initiative that is the one that she will be talking today, for the creation of i-BALSAC, a multi-sectoral platform for cutting-edge research on biomedical and social science research. So welcome very much, Helena, for coming down to the south here to Andalusia. And as usual, we have 45 minutes to 1 hour talk, and then we open the floor for questions.
Thank you for being here. Thank you, Diego, and all the other organizers for giving me this opportunity to be here in Sevilla and to talk to you about the i-BALSAC historical demographic database, with a special focus on its use in health-related research. So this is the way I have organized my presentation. I will try to answer the following questions. First of all, what is i-BALSAC? Not very original, but necessary, I think. A few words on the specificities of the Quebec population for research in genetics and health. Then I will talk to you about what type of health-related research is conducted with i-BALSAC and how it has evolved with time, because as Diego mentioned, it started a long time ago. Then a few words, not too long, because this is really not a technical presentation, but I will talk to you about how do we select the subjects, how do we go about data access and privacy concerns to deal with the database. And finally, this new project we are just starting, so what is currently being done to support health-related research with i-BALSAC. But before I start, I would like to tell you that I prepared for this presentation from two standpoints. First, as Diego mentioned, I've been using the i-BALSAC database for more than 25 years now, since I started using it during my doctoral studies when I was working on Alzheimer's disease. And ever since, I've been doing research in historical demography, but also health-related research that we sometimes call genetic demography. Also, since 2010, I am head of i-BALSAC, so that means that together with the team, I can make decisions on the orientations of the database, so I will share some of this with you too. Last thing before I start, I really hope that some of what I will say will resonate to you, you will be able to make links with your own work and preoccupation. And at the end, don't hesitate if you have questions. I will be happy to at least try to answer them. So let's start. What is i-BALSAC? I will give you a very brief history of the database. I will provide you with an overview of its content, linkage process, and structure of the database, and a few words on the sources for the data, their quality, their exhaustivity. There are so many things I could say, because as Diego mentioned it's 50 years of history that I will touch many topics, but won't go too deep. Okay, what is the raw material for the construction of the database? In Quebec, we are quite lucky because the birth, marriage, and death certificates that were recorded by the Catholic priests since the early days of French settlement in the 17th century are of very high quality, very complete, and remarkably well preserved. So using this resource, the construction of 2 databases were initiated slightly more than 50 years ago for the first one, which is called the Registre de la Population du Québec Ancien. It's at the Université of Montreal, and basically they have been working on the 17th and 18th century. And the second one, which will be 50 years old in a few years, is called the i-BALSAC database and is at the Université of Québec in Chicoutimi. A few more words specifically on i-BALSAC. So the construction was initiated by a historian. He's called Gérard Bouchard, just retired a few years ago. But back in those days, he was just back from France, where he had done a PhD on social history, and he had learned all the techniques of family reconstitution that were devised by Louis Henry. So when he came back to Quebec, he wanted to apply these techniques. But up until then, they had been used only on small villages or one parish, and he really wanted to work on a whole region to be able to deal with issues like migration and things like that. So the first phase of the database lasted about 15 years, and it was the family reconstitution for the Saguenay-Lac-Saint-Jean region from 1842, which is more or less the onset of French-Canadian settlement there, to 1971. Maybe I will go, I put a little map here just to situate you. So here, of course, you have Quebec, but as you know, 99% of the population lives in this area, which is here, lives along the St. Lawrence River. Montreal is here. Currently, about half of the population in Quebec lives in the greater area of Montreal. The whole population is 8.4 million. Quebec City is here. And the Saguenay-Lac-Saint-Jean area, Saguenay River, Lac-Saint-Jean here, hence the name of the region. So in this, and the Université of Québec at Chicoutimi is about here. So that's the reason why the work started in this region. And it was, as you noticed, populated later than the regions along the St. Lawrence River. And since the beginning, it has represented more or less between 3 and 4% of the Quebec population. So that was the first phase. Second phase, which lasted about 20 years. So at the end of the 1980s, the decision was taken to expand the population database to cover all of Quebec, all regions. Another decision was made, though, for financial reasons, but also because by that time, the database had really started being used for genetic and medical research. So the decision was taken to restrict entry to marriages only, no birth, no death, only marriages, because these marriages were really the basis for the genealogical approach that is used in human genetics and health-related research. So altogether, about 2 million records were entered for the period 1800 up to 1965. And very quickly, because I never talked about this, but I realized that often at the end of my presentation, people ask me, where does the name i-BALSAC come from? And so it's the name of the first, all these regions are located in the eastern part of Quebec. So when they started the expansion of the database, they were working on these regions, and I suppose somebody was playing with the first letters and came with the idea of calling it i-BALSAC, except it's a S, not a Z. Okay, as I said, no technical details, but I want to tell you a little bit about the construction and the structure of the database. So first, of course, all records must be transcribed in a database format. So we go from records like this, and we enter it in the database. And up until now, because I will talk again about a little bit about that later, but it has always been research assistants who were reading the records and transcribing them. Then, if you want it to be a population database, you have to link these records together. So the basic unit of information in BALSAC for linkage is what we call, it's a translation, but couple mention. So that means that comprises forenames, first and last name of the husband and of the wife. So yeah, nominating elements. And the decision-making process for linkage draws exclusively on nominative information contained in the records, plus we have a whole bunch of coherence routines for dates and things related to the family structure. And we do the linkage in a chronological order. That means that normally when we enter a new couple, the parents of this couple should already be in the database, and so we can automatically link them to what is already in the database. And the linkage system aims at grouping in the same family all the mentions appearing in the records and referring to the same couple. So don't worry, we won't read this into details, but just to tell you that each line corresponds to a record. So this couple here appears either first at their marriage, and then if it's the Saguenay region, you can have birth of children, death of unmarried children will be in this file too, or marriage of children, and eventually their own death, and even remarriage sometimes. And by bringing all these families together, links are established between generations, and eventually the genealogy of the whole population is reconstructed. Very quickly, database structure. We work with SQL Server. We have 3 main tables for the data. We also have various other tables, including codes for occupation, residences, and we have names and surnames in their original form, but they are also phonetized for linkage purpose. Quickly, for instance, in a marriage record like here, you can have up to 7 geographic variables. So all this data is coded in a dictionary. So as of last year or something, no, I don't think, we had approximately 22,000 different names of places in the dictionary that correspond to a total of 12,000 distinct localities, and they can be grouped in different geographical sites, like region, country, province, if it's Canada, state, if it's United States, etc. We've also coded all the occupations. So in a marriage record, you can have up to 6 occupations, although as you can imagine for women in older days there's less. But almost 100% of occurrences were coded. We use the HISCO and HISClass codes that have been devised in the Netherlands and are largely used now in historical social sciences. Okay, so BALSAC today, roughly, is 2.2 marriage records for Quebec, plus 550,000 birth and death records for the Saguenay-Lac-Saint-Jean region. It's a longitudinal database that allows for automatic reconstruction of family histories, geological ties, and kinship relations among individuals. It covers all regions of Quebec since the beginning of European settlement in the 17th century, because I will come back quickly to it later, but we've traded data with the Montreal database, so we also have access to their data. So we can automatically reconstruct the genealogies over 4 centuries. Again, not too many details, but I thought it was important to mention to you that roughly during the French regime, 17th, 18th century, approximately 100% of the population was French speaking. And you could say 19th, 20th century, it is roughly about 80% of the population, which is of, I put French Canadian / Catholic origin, because of course the records are religious, but there's really a correlation between French Canadian Catholic, although there were also some people of other religions like Italians, maybe even a few Spanish who came and were also Catholic, and English-speaking population and Protestant records. And Protestant records, we don't really have time to talk about that, but their quality is a lot lower. The demands of the Protestant priests of the Protestant church were not as precise as the one from the Catholic Church. So very often for the older period, it's very difficult to link them. So we have really concentrated on the French Canadian population, but progressively we're also doing the other denominations. Research with BALSAC, what I want to mention to you is the database is used extensively, and it's really a strong characteristic of the database. It's used extensively both for social and for biomedical research. So that means that multidisciplinarity has been really a major feature of the history of BALSAC. So for instance, from last year, there was 45 projects ongoing, and you can see that they really belong to different disciplines. And lastly, although BALSAC was developed and is still maintained at the Université du Québec à Chicoutimi, it is the joint propriety of 4 universities, 4 Quebec universities. Okay, BALSAC. Second part, not very long, but I wanted to tell you some specificities of the Quebec population that have really given orientations to the research that has been and is still conducted. So I will tell you a few words about founder populations and founder effect. I don't know if it's things you know already, but it won't be long anyway. And very few words, if there are historians in the place, I apologize, because it's going to be a very, very short Quebec settlement history. What is a founder population? So a founder population is a population that is derived from a relatively small number of migrants who leave what we call the mother population, and the immigrants are there, so they leave the mother population, and let's say that the colors here all represent different genes. So since it's only a part of the population that leaves the mother population, it's also only a fraction of the gene pool, all the genes that you found in the population that will leave by chance. Some genes will be overrepresented among founders, some will be underrepresented from the previous frequency, and some will be just absent, like the little turquoise or something. And then they come, they migrate, and they form a new population. You can think, for instance, France, Quebec in the 17th century. So, after that, time goes by, generations elapse, and you see that after some time, the new population has a gene pool that is quite different from the population of origin. And there are some genetic consequences of the founder effect. Founder effects are very important in population genetics. They have been studied in all sorts of manners. So what has been observed is that you have a loss of genetic diversity. You can see there are a lot less colors here than there were here. There are changes in gene frequency, and there is an increased frequency of some genetic diseases. And this is often like that founder effects have been observed in population. It's first clinicians who observe an increased frequency of some genetic disease, and then if they know a little bit about the history of the population, they might suspect there has been a founder effect. There are many founder populations. I just have a few examples here, but there are many others. Sometimes it's for religious reasons, like these religious groups here who migrated from Europe in 17th, 18th century, in 19th century, I think, in the United States, French Canadians. There is a founder effect in Costa Rica, Iceland, a lot of people know about it, Finland, Afrikaners in South Africa. And these are called young founder populations because they are less than 50 generations old. But for instance, here in Europe, in mountain valleys, on islands, you find a lot of founder population, except that sometimes they are a lot older than that. Medical and population geneticists have a special interest in founder populations because of the characteristics of their genetic structure. And as I told you, the Quebec population is a founder population. Why? This is where I give you the crash course in the population history. We talk about the initial founder effect. What we mean by that is we talk about the first European immigrants who came in the early 17th century. The French rule or regime lasted for about 150 years. And then we had the British conquest. we say that there were about 10,000 pioneers. A lot more people came, but some of them left, some of them died without leaving any trace. But and of these, about 8,500 experienced the family life and had at least one child. So in genetic, that means that they have some probability of having transmitted their genes to the following generations. The vast majority of these immigrants were of French origin, not all, but the majority, and we have been able to calculate that they account for between 80, these founders here, they account for between 80 and 90% of the gene pool of the 6 million Québécois who are of French Canadian origin. These dots represent the opening of Catholic parish registers, so just to show that at first everybody was along the St. Lawrence River, and then progressively after 1760 there was an expansion of the population to occupy new territories, including here the Saguenay-Lac-Saint-Jean. So after the British conquest, it's virtually the end of French immigration, and the French Canadians lived in relative isolation because of geographic reasons, for instance, those who were here, but also because of religious factors. Because although there were new immigrants, you know that the Catholic Church didn't really encourage mixed marriages, so there were some, but they were quite limited. There was very rapid population expansion. Maybe you know that for a long time, French Canadian had a strong reputation for very high fertility. Although since that time, some authors have shown that it's not that typical of French Canadian. It's typical of all new populations who have a lot of land ahead of them and they know they can settle their children, so they can have big families. The last thing I want to tell you, there were new regions that were progressively open to settlement because of land shortage in the St. Lawrence Valley, so that includes, for instance, the Saguenay-Lac-Saint-Jean area, and this has led to regional and local founder effects. And I know time always goes by faster than we think when we present, so I don't want to spend too much time, but I want to mention you that one of the strongest regional founder effect is precisely the Saguenay-Lac-Saint-Jean region. So this region has a very specific profile of inherited disorders, and it explains in part why so much research was conducted in that region. And just like all founder populations, there are some specific, all populations have genetic disorders, but in founder population they have specific profiles. So here you have a list. It looks terribly discouraging when you look at it, but it's like that in all populations. A list of genetic disorders that are found in the French population, French Canadian populations. But you could find in the literature the same tables for Iceland, Finland, Okay, third part of my presentation. What type of health-related research is conducted with i-BALSAC and how has it evolved with time? I will tell you that basically 3 approaches are used to conduct research, but I will also very quickly tell you that the genealogical approach is by far the most widely used, so I will really focus on that one. And I will give you examples of 4 main themes of research: genetic epidemiology of inherited disorders, genetic epidemiology of complex traits, and a few words on population genetics and evolutionary biology that are less but also related to health research. So the 2 first approach, I call them working with individual biographies or working with family and intergenerational approach. So in these studies, the outcomes are variables that are related to death because you can find the information in the database. For instance, there has been work on age at death, longevity, infant mortality, or sometimes some health scientists work with this approach, but using a cohort of individuals affected with a specific disease. To conduct these studies, you want to have information on birth and death. So up until now, because this is also changing as I will tell you later, these studies have been conducted on the Saguenay-Lac-Saint-Jean area since, you remember, this is the only area where we have the complete civil records. Okay, statistical methods to do the analysis, and sometimes also the use of genetic analysis can be used for to study the intergenerational transmission transmission of traits. Quick example, because I brought many examples, so I cannot spend too much time on each. But a paper by a demographer at Université de Montréal, which, of course, I chose many examples where I was involved because it's easier to talk about them. So that was a study on age at death between marriage and after marriage, And we wanted to see if there was a link between socioeconomic status and actually when he was looking for a gradient in the Saguenay-Lac-Saint-Jean region. So I'm just showing you this so that you can see the approach. And no, we didn't find anything except that the mortality was lower among farmers, so it probably had to do with a rural-urban difference. Another example, this time of intergenerational study. That was some years ago. We conducted a study on, we wanted to see if there was a link between age at death of individual and age at death of their parents. So we selected individuals who died after 90 years old. And we formed a control group of individuals, I wrote it there, okay, who died between 50 and 75. And I hope you can see, but you see here, we look at the mean age at death of their fathers and their mothers, here for the subject, here for the controls, and you could see that yes, among the subjects who died at 90 years and older, on average, their parents died about 4 years later than the controls. So this is the kind of study we can do. Now, with what is mainly the most original component of the research we do, the multi-generational and genealogical approach. As I said, it is by far the approach that has been used the most for medical and genetic research done at BALSAC. It can be done just using only data from BALSAC or in combination with medical or genetic data. And why is it of interest? Well, quick explanation. Here you have the genealogy over 3-4 generations of an individual, but the genealogy of a population contains information about, of course, the ancestors who participated in the reproduction and the kinship links that connect these ancestors together. If you think about it from a genetic perspective, it therefore contains the individuals who have transmitted their genes and the transmission path of the genes are here over the generations. So, as I said before, the genetic pool of a population, that is the sum of all the genes that you find in the population today, is made up of all the genes transmitted by these ancestors. And that means that it is shaped, this gene pool, by the demographic behavior and characteristics of these ancestors. And this is why, and I think I will say it again at the end of my presentation, I think demographers in general and historical demographers in particular can make a very interesting contribution to health research. Okay, quick history. So the genetic research started not very long after the beginning of the construction of the database. And this is when Gérard Bouchard, working with, he developed a research program in medical and human genetics, and he was working with medical geneticists and physicians because, of course, as I said, demographers can contribute, but they cannot do it just by themselves. You need to work in collaboration with health scientists. So over a period of about 10 years, most work, again, was conducted on the Saguenay-Lac-Saint-Jean region, but in 10 years, there was about 30 research projects that were conducted on 16 different genetic diseases. And the main themes of these research were to document the characteristic of the introduction and the diffusion pattern of inherited disorder. So at first, the research was really on inherited disorders, and people wanted to understand how were the mutations, well, it was not even the mutations then, how were these disorders introduced in the population, and how did they disseminate on the territory. And they wanted to understand the elevated frequency of some of these disorders, Often it was trying to figure out if it had more to do with the founder effect or if it was something that had to do with the demographic behavior or history, namely inbreeding. And they published many papers. I know because I was starting my PhD then and I had to review them all in order to do my work. And I think it's worth mentioning two things. They also published a book which unfortunately was never translated in English or any other language, but it's really a book on the population genetics of the Saguenay region, and it's very, very interesting. And I also want to mention that they really had, it's a bit of unrelated to my presentation, but I think it's important. It was very important to them to do things for the population, because the population in Saguenay is aware that there is this problem with the genetic disorders. So they also published a book called Why Inherited Disorders, where they were trying, in words that people could understand, to explain all the historical factors that led to the situation, because there is a lot of guilt associated to the presence of these disorders. People wondered why in my family did you do something wrong? So with the science, sometimes often you can diminish the guilt and help people to understand. And if you understand what happens, it doesn't solve the problem, but at least you feel empowered a little. Okay, I also told you I would say how each theme of research has evolved. So for inherited disorders, I would say that since that first period, progressively all the genes linked that were causing these diseases were identified. The mutations that were specific to the Saguenay region, for instance, were identified. So we have been able to conduct studies that were more precise because we could group individuals according to the mutations they were carrying instead of only their phenotype, that is their disease. The other thing is that progressively, and that was a good thing, the studies were expanded to other regions or localities of Quebec, and that was good because the counter effect of doing all this work in Saguenay is that there is some sort of stereotype that developed. A lot of people, even today in Quebec, they think that there are so many genetic disorders in Saguenay, and no, there is not necessarily more. They have a specific profile of disorders, but nobody has been able to prove that they have more. And use of increasingly sophisticated statistical methods. I think it is the case for many sciences in the past 30 years. A lot of methods have been developed and applied. Quick example, this is one of the first paper, I think, on a disorder that's called myotonic dystrophy, and Saguenay has the highest frequency in the world. It's a dominant disease. And Claude Laberge, who is the first medical doctor in Quebec who did a PhD on genetics, and his thesis was called, I think, The Genetics of French Canadian. He went to Johns Hopkins University, and so anyway, he was quite active for many years. I'm just showing you this because of course, to us now it seems quite a very simple approach. You find people, you reconstruct their genealogy, you try to find common ancestors, but they had no control groups, so the results, they always end up with the most frequent ancestors, which is quite obvious. But anyway, that was the beginning. Then, as I said, there were diseases that were studies that were found in other regions. This one I just want to show you because I think it's interesting. This disorder here, Leber hereditary optic neuropathy, it's a disease that is transmitted through mitochondrial DNA. And again, maybe everybody knows here, I don't know, but mitochondrial DNA is transmitted from mothers to their children. So we all received it for our mother, except that only women transmit it. This is why it's fantastic to study disorders like that with genealogies, because it's the genes are transmitted, the mutations are transmitted only through the maternal lines. So we can be absolutely sure that the woman who introduced the gene in the French Canadian population in the 17th century was identified. So this study was conducted by a geneticist. And she also studied the migration, if I may say, of the mutation on the territory. By chance, the ancestors moved from Quebec City and progressively along the St. Lawrence River. And this is currently where you find the different cases in the population. And it's a disorder, if it's not treated, you can become blind. And last example, it's a very, very recent study, just to show you how things have evolved. It was published in December 2018 in the American Journal of Human Genetics by a PhD student working with a geneticist mathematician at McGill University. And he was working, it's always incredible names, so I'll let you read it. It's chronic atrial and intestinal dysrhythmia. Anyway, it's a rare recessive disease, and some mutations are found in Quebec. So he was working on a specific mutation and using quite sophisticated statistical methods, he was able to pinpoint two families or two couples that might have introduced the mutation. And using his statistical method, he can also measure an expected mutation frequency in the regions of Quebec. And this is really, I will come back to it in the end, a step towards the idea of combining genealogical and epidemiological studies to design, for instance, carrier, disease-causing allele carrier programs, etc. Complex traits. So starting at the end of the 1980s, beginning of 90s, there was more and more interest to understand the genetic basis of complex traits. But as you can see in the name, they are complex traits, they are very complex to study too, and they are even more complex than what people were expecting, I think, at the beginning of the 90s. Because when I look at my own thesis that I was doing in this first wave of enthusiasm for combining genealogical studies and complex traits, it was very interesting to do, but difficult to obtain results that can really be applied in the... But anyway, the interest was to gain a better understanding of the role of genetic factors in the etiology of these disorders, and this has been working, contribute to the study design of gene mapping projects by reducing or taking into account heterogeneity of genetic background and familial factors. It has proven to be so complicated to identify the genetic factors involved in many, I'm talking here about cancer, hypertension, disorders like that, where Alzheimer's disease, where we know there is familial aggregation, for sure there are some genetic factors, but they are very difficult to identify. So if you know well the population from which your cases come, you have more chances to identify the genetic factors that are really specific to the affected individuals and not only characteristic of the whole population. For many of these disorders, I will give you in 1 second an example for breast cancer. When the research started, it didn't take too long to identify genes that were responsible for specific forms of these complex traits. Normally they're very rare genes, but they have a very high penetrance in families that are carrier of these genes. But as I said, on the whole approach has proven very challenging. On the other hand, I think it's one of the fields where the future is the most promising to combine genealogical studies, historical demographic databases, and the study of these traits. So this is something I did during my PhD thesis. So I just want to say the objective was to detect then some cluster of patients related to common ancestors and presenting a level of kinship or inbreeding that was higher than observed in the unaffected population. And also because we knew already there was a lot of heterogeneity in the disease, we thought that maybe some of the factors that we investigated, like we worked on age at onset, carrier or non-carriers of a certain allele that is known to increase the risk for Alzheimer's disease. So we were doing genealogical analysis on that. Then a few years later, I worked with a group that's the example on breast cancer, and yes, you might know too, but there are two major genes for breast cancer, BRCA1, BRCA2. And in founder population, remember we said there is more genetic homogeneity. So some mutation, there are not more of this form of breast cancer in Quebec, but in the families that are affected with this form of breast cancer, there is more homogeneity among the mutations that are found. So we studied a founder mutation where we had 17 families that were carrying this mutation because we wanted to understand, again, the diffusion pattern in the population, maybe find who could have introduced it. Of course, it doesn't change anything to the people who are affected, but it's interesting to understand how it might have entered the population. So we are pretty sure we found a couple. I say the couple because we can't know if it's the man or the woman who introduced the mutation on the Quebec territory. But this time, it's not a mitochondrial disorder. It's on a autosomal chromosome. So you cannot be 100% sure. But the more cases you add, the more it's the probabilistic approach, I should say. But I just want to say before I move to the next slide that the colors here, they represent the region of marriage of all these individuals, and these are all the ancestors that connect this couple that we found and the affected individuals here. So you can see that there is most likely a regional concentration of the mutation. And I say most likely because I really want to insist on that. You can never take the result of a genealogical or demogenetic study and directly transpose it to epidemiology. You really have to work with epidemiologists combining it with an epidemiological approach before you reach that type of conclusion. And this, quickly to mention to you that the approach is still used. Recently, a colleague of mine was working on drug intolerance. I didn't even know what it was before I was working on it. So it's people who are affected with hypercholesterolemia, and they have a mutation on a specific gene, and some of them have intolerance to the drug they should be taking. So they wanted to see if this could be related to the major effect of a certain gene. So they performed a genealogical study to find out. The results were not conclusive. Quickly, to conclude on this part. So, studies in population genetics. There is also an interest for consequences of settlement history, founder effect, but other demographic events on the genealogical and genetic structure of the population. And then when I say genetic structure, I include disease-causing genes. And these studies have also evolved with time. So at first, again, they were in the Saguenay region, but then we were able, the more BALSAC developed, the more we were able to study the regions and conduct comparative studies. And I would say for the past 15 years, we've also started doing population genetic studies, combining genealogical and genetic data, because before it was impossible to get genetic data for population genetics, impossible in the sense that the costs were so high that you could not have big groups that you need for population genetics and get genetic material on them. But progressively the cost in molecular biology went down and it was possible to start that type of study. So, two quick examples. Many years ago, again with my colleague, we did a study on consanguinity in Quebec, again to try to contribute to dissipate this myth that there is more consanguinity and more inherited disorders in Saguenay-Lac-Saint-Jean. So we were able to measure consanguinity in all regions of Quebec, and they're placed here more or less into a west-east gradient. So you can, I want to go quick because I realize the time goes by, but the darkest part, let's say, is the part of the consanguinity that is due to population history, and the light one would be the close consanguinity, that is the consanguinity that is due to marriage with somebody you are related to, like the choice of your spouse. So we could show that here in Saguenay-Lac-Saint-Jean, the close consanguinity was certainly not higher than in any other region. And yes, in the eastern part of the province, because of population history, population settlement, the general consanguinity is higher, and that explains in part why they have this specific profile of genetic disorders. And then some years ago, we started working with a genetic epidemiologist, Marie-Hélène Roy-Gagnon, and a geneticist, Daniel Arruda. And one of the things they did is that they compared genetic data from from the cohorts of individuals that we recruited, and these individuals were from the general population. They don't have any specific disorder, and the genealogical data. And without entering into any detail, you can see that what is really, really nice is that if they did the analysis with the genetic data, with the genealogical, and look, each dot corresponds to an individual, and the color is its region of origin. So you can see that the genealogical data can provide is very similar to the distribution with the genetic data. So it was a confirmation of what we thought, is that there is a very strong link between the geological structure of a population and its genetic structure. Last, quickly, there were some studies in biodemography, mostly on intergenerational transmission of demographic traits like infant mortality, fertility, and in that type of study, we always say that i-BALSAC then becomes a sort of a human population laboratory. And progressively, what we have seen is that these studies have gone from studying demographic characteristics to evolutionary phenomena. In the past years, there's more and more evolutionary biologists want to use the data to study, for instance, on selection. And you might think here that we're a bit far from health-related studies, but we're not that far, because if you understand all these population genetic processes, you can better understand the genetic component of the disorders. Again, example, a study I did with my colleague many years ago, but I always say that I'm sure in my whole career that will be the most cited study, but at the same time, it's for a demographer, it's probably very simple. We used to go to genetic meetings, and geneticists are often very interested in calculating the age of a mutation, because it gives them information about their diffusion pattern, where they might have originated from, and they always use 20, 25 years at generation length, and we thought, oh, it's too short. So we calculated it, and we were able to publish in a genetic journal these figures. So we told them that the generation was closer to 32 years, and we said that the population we were using to measure this was a natural fertility population, so we thought it could correspond to what was found over a long period of time in human populations. And yeah. And more recently, there was a Swiss, again, evolutionary biologist that was also working on the Saguenay population, but with no specific interest on population. It's somebody who is working on population expansion, and he's comparing individuals who live on what he calls on the wavefront, so people who are moving to occupy the territory, and those who remain in the core, the range core, and he compared their fertility, age at marriage, and he was able to demonstrate that people on the wavefront had one more child, but still over a long period of time, on average, it's important. And also, that was a big result. It was that they also had more married children, so that means children who might have their own children, and that there was an intergenerational correlation of this factor on the wavefront, but not in the core. And this, again, if you think about it over many generations, it can have a very strong impact. So again, it seems like a quite simple study, but he managed to publish his paper in Science, so I think he convinced some people of the impact of the study. Okay, so evolutionary research from i-BALSAC standpoint, and I'm getting to this new project that we are just starting. So over the years, we went from very focused studies to more exploratory studies. We went from researchers who were working on small data sets, genetic disorder, between 10, 15 patients, controls, to very large data sets to explore their new hypotheses. From analyses that were performed at i-BALSAC, people from the medical world, often they would ask us to reconstruct the genealogies, do the analysis for them, and send the results. It's not like that anymore. Now they want us to extract the data sets, send them the data, and they will analyze it. And we went from an almost exclusively genealogical approach, based on marriages, to a strong interest for individual and familial demographic characteristics, that is, including birth and death. I will go quickly because I want to go to the new project. Is it okay? So I thought, again, I said it was not a technical presentation, but I need to mention to you a little bit how we proceed to conduct this analysis. So what I wanted to tell you is that how do we go for the selection of study subjects? So if we want to have a sample from the population, well, if we're going to do a genealogical or biodemographic study, that means that contain no medical or genetic information, we can select directly in BALSAC. If we want to combine the genealogical and the genetic data, the genealogical and genetic data, but we are working on what we call neutral DNA variants. So variants that are not related to any specific disorder, then, and we have done that, you can recruit directly in the population. We went into certain communities and said, are you interested in participating in our study? And people volunteered to participate, and that's fine. Of course, they had to sign a consent form, and our project had to be approved by our research ethics board. On the other hand, as soon as we work on what we call patient samples or affected individuals or individuals or carriers of disease-linked mutation, that means that the recruitment has to be through some medical research project. And we have to collaborate with health scientists who are then PI of the principal investigator of the project, and all the ethic approval and consent form has to go through them. And if, in order to be able to reconstruct the genealogies, we collect family information to be able to locate their, normally if we have information on parents or grandparents, then we can connect to the database, and we've designed our own questionnaire. I just put it there to show you, but of course we won't look at it. And we always try to improve it, because throughout the years we've seen what people understand well, less well, because we try to get the most precise and accurate information possible. So this is how it works. We receive the genealogical data from the participants. Any genetic or medical information should be transmitted only if relevant and will not be integrated to the BALSAC register. I'm reading this because you will see in a few minutes that it is something that we are changing now, so it's a big change for us. Then we reconstruct the genealogy because it's done mostly automatically. It takes between 5 and 10 minutes to do the genealogy, and normally we go back up until the first ancestors of this person came to Quebec. And then we can transmit either the data or the result of the genealogical analysis to the researcher. And at the end of the project, we destruct all the information that linking a participant to the project. We only keep the genealogical information. So this is how the framework in which we have worked traditionally, and we offer researcher support to do all these operations. And we have 2 access procedures. One is for what we call non-confidential data, so people who want to use only data that is from the public domain, and people who want to access confidential data. And as you can imagine, as soon as you have contemporary data with names, medical information, genetic information, you fall in the confidential process. Okay, what is currently being done to support health-related research? As I said, in 2010 I became director of BALSAC. We made decisions on because every 3 or 4 years we write a new proposal to continue the development of the project, and so we have to decide what are our priorities, because we cannot do everything. So this is the priorities we set for ourselves a few years ago. First of all, we decided we are up to 1965 for marriages. We decided to stop this chronological integration of marriages for a little while, because with the genealogical questionnaires, as I said, normally if we have parents, grandparents, we can connect to the database. Also, I don't know how it is here, for instance, in Andalusia, but in Quebec, starting in the 70s, people married less and less. So for sure, we cannot relate only on marriage records anymore to reconstruct the population. And as I said, there was more demand from the researchers to have complete civil records, including birth and death. So we decided to work on that. We also wanted to be able to integrate other types of data in the database. So we adapted the database structure to allow for that. We also wanted, I don't think I mentioned it at the beginning of my presentation, but PRDH has been working for 50 years, the development of their database. We have been working a long time. We have census data from another research group, but we all work independently. So we thought it was time to establish a formal partnership and to bring our data together. And also we wanted to improve data access. As I said, up until now, it has always been researchers ask for data. We send it to them after they have the necessary approval. But we wanted, for the non-confidential data we have, we wanted to provide a direct access for research because, after all, our mission is to develop an infrastructure that's used for research. It's just not for the sake of development. So we had a first project that we called IMPQ. You have the title there in English, but the French acronym is IMPQ. And it lasted about 5 years, and it's really about the one we are just starting that I want to talk, so I will go quickly over this one. We merged the BALSAC and RTQA databases so that now researchers can access one infrastructure with a common format going from the 17th, to the 20th century, and we also integrated new birth and death records. Then there was also some work done on census data, and we developed a linkage program to connect census data and BALSAC families and to connect censuses together. So we made longitudinal data using censuses. And so we developed a program to do that. Yeah, and I wanted to say that in North America, we don't have population registers like many countries in Europe, for instance. So for us, it's incredible the power it brings to our studies to be able to combine these two types of studies. And actually, we're not the only ones in North America who are doing it now. And we developed a portal to access this data. Now, this project was being almost completed. It was time to think about our priorities, to define our new project. So we decided on 3 things. That we wanted to continue the entry of birth and death records to reach the 20th century. Also, and this is very, very major for us, we wanted to design a formal and sustainable structure that would integrate the genealogical and the genetic data. Because currently, and I've mentioned it, but maybe you didn't notice, those data are only linked during the time of specific projects, and at the end, we cut the link. So researchers can never go back to the same data combined together. And also, that was also something we had been thinking about for a while. We wanted to take advantage of some of the enormous potential offered by geographic information systems, and I think here there are many specialists of that. But so we wanted to create a framework to analyze and interpret the spatial characteristics of the genealogical and the genetic data. So we are very happy because the project was funded. When we design major projects like this, for us, the main agency where we can apply for money is called the Canadian Foundation for Innovation. So it worked. We are very, very happy, and the project is starting really this month. We had the first team meeting at the end of May. So quickly, I will describe it to you. It rests on 5 objectives. The first 3 objectives has to do with genealogical, genetic, and geographic data that will be assembled, standardized, and integrated into the new infrastructure, which for now, we might change the name, we have called i-BALSAC. And to facilitate the exploitation of these data, because again, it's great to generate new data, but people have to have access to the tools to be able to analyze them, we will develop again spatial analysis tools, historical GIS, and statistical methods will be developed. I go back to each of these objectives. So integration of demographic data, I'm showing you this table. I would like you to look at the red line, and specifically at this number here. So we will enter 6 million records in the 3 next years, because the project lasts 3 years. You can look quickly, that means more than has been entered in the both population databases in Quebec over 50 years. We have a partnership with the National Archives of Quebec, who are providing very good quality images to do the work. And why very good quality is necessary? Because for the first time, we have decided to work with handwritten text recognition technology. Well, I put some information here, but I don't think it's on the intro for now, so I will skip it. But once the data is in the database, we will do the linkage, automatic linkage at that time. For genetic data, in fact, we won't produce new genetic data. The goal is to integrate into a single set the genetic data that has been collected over the years on individuals that were recruited in the Quebec population. And this will be achieved in two ways. First, collaboration with a number of biomedical scientists who have authorization from the participants to share the genetic and, when possible, the genealogical data. And this is what I want to tell you a few words about. We have a partnership with a project that has been going on for a few years in Quebec. It's called the Cartagene Project. For those of you who know about the UK Biobank, I know it has a lot of similarities. And quickly, it is the largest population cohort of Quebec. They recruited 43,000 individuals aged between 40 and 69 at time of sampling, and they have collected biological samples, health, lifestyle, socio-demographic data, and they have also collected, for those who wanted, the genealogical data. So through our collaboration with Cartagene, they will share with us the genealogical and the genetic data they have, plus they will harmonize their portal so that for researchers it will be easy to get information from the i-BALSAC portal, that is genetic, geographic, demographic data, and the medical and health and epidemiological data from this database. And I think this is very, very exciting. I think it will give way to a very interesting project. The geography, again, it's quite standard, so we'll go quickly, but we will establish a correspondence between the geographies of the Quebec Vital Registration and of the Quebec component of the Canadian censuses. And we will characterize the local spaces from a demographic and socioeconomic point of view to contextualize the genetic and the genealogical data. I won't read it, but this is the kind of data that we will have. And this also I am very happy because we have known for a long time that these data exist, but there had been no consistent and systematic effort to digitize and enter them in databases. Part of the work is done, but it's at different places, so now we'll group it and build the HGIS. Genealogical information can be used to improve genetic research in many ways. Here are 3 examples. It can improve the methods for imputing missing genetic data, and my next slide is an example. For estimating haplotypes and genomic segments shared either among individuals or within a single individual. It can improve methods for genetic linkage and association studies, but there are few statistical and bioinformatic tools that can exploit very large genealogical structures. In this example here, it's actually from a small population, small isolate in Holland, but I always thought this figure was excellent to show there are not so many people at the bottom here, but because of the structure of the population, and I suspect the Quebec population structure looks a little bit like this, you see how complicated it gets. And we will develop statistical tools to promote the type of analysis with a special focus to optimize the joint exploitation of genealogical and genetic data sets. Quick example, you have again an example based on mitochondrial DNA because it's easier to show. Here you would have this woman was genotyped, so you know, for instance, her haplotype or haplogroup for mitochondrial DNA. Automatically, she received it from her maternal line, so you can go back to, for instance, this common ancestor. And from there, you can impute using the available genetic data that you have and bioinformatic methods. You can impute, project the genetic information in the ascending lines of contemporary individuals and impute it in the descending line. That means all the individuals in red, if you know, just by knowing this genotype here and using the genealogical data, you can impute even if you don't have the actual genetic material for this individual. So, and you can also have methods for autosomal genes, just a lot more complicated, so I don't think I could explain it properly. But thus, the goal is to trace a portrait of the genetic structure of the population and then provide some, it's very interesting for all geneticists. The more you know the genetic structure of the population you're working on, as I was saying earlier, the more efficient you can be in your approach to study your topic of interest, a specific disorder or mutation or whatever. And finally, we will develop a portrait. Of course, this work will be done with a team. I just want to say there's historical demographer, historical geographer, historians, population geneticists, medical geneticists, genetic epidemiologists, and their teams from their labs or research group. We also have partners. It was really a project. It's the most complex project we have designed so far. And this is why one of my last slides is to tell you what are the main challenges that we expect. Well, the first one is working with handwritten text recognition. It's very, it opens up incredible possibilities, I think, but it's relatively new, so it's a challenge. And we know, of course, as I told you at first, that so far it has always been research assistants who are reading and entering the data. Of course, we cannot expect to reach the same quality, but we thought it was worth it in view of the number of data we wanted to enter. Integrate the genetic data. What I want to do is that it raises quite big confidentiality issues. It will be necessary, of course, to adapt i-BALSAC. As I told you, that up till now, we never kept the genetic data. But I think the mentalities are ready for that. And finally, the diversity of researchers, personnel and partners involved. Of course, it will be a challenge because we really want people to work together. We want real interdisciplinarity. And so as we are project leaders at i-BALSAC, it will be our responsibility to make sure that we all focus on a common vision for the project. Okay, this is my one before last slide. In summary, what I think is and can be, because it has not expressed, I'm sure, its full potential yet, the scientific contribution of health-related research conducted with demography, using demographic historical data. Well, in the context of population genetic studies, it can help to increase the knowledge about the context of introduction, transmission, diffusion on the territory of genetic variants, including that can be associated or not with diseases. In the context of epidemiological studies and clinical practice, it can really contribute to improve public health through better predictions of incidence rates. This is what I was trying to show you with the very recent study on the inherited disorder. The screening and prevention programs that are targeted in specific geographical regions, or it can be specific ethnocultural communities, or in the context of molecular genetic studies, it can help for the identification of genetic determinants of health. I tried to tell you in particular how complicated it proves to be with genes associated with complex traits. I also told you a few words about the possible social contribution to understand, to demystify, to remove guilt, in particular in areas or in communities or in groups which are confronting elevated frequency of some inherited disorders. And my last slide, I wanted just to make a few, share with you a few personal thoughts. I think, as I just said, this potential is still to be brought to its complete expression. Also, of course, I've been telling you the context I know, which is the context of the Quebec population, and each population has its own population history, issues of data availability, research agenda. So I'm presenting you an approach, but of course it has to be adapted to each population. On the other hand, you have seen at least partly, how health-related research with the historical data, it has been conducted in Quebec for 40 years, and it has changed a lot, it has evolved with all sorts of factors, but it's still there, and it's continuing, and I think it's even increasing, because new questions are emerging. You can think about epigenetic, you can think about genetic genealogy, and again, genetic predisposition to chronic and degenerative diseases remain a major concern, especially in society that face the aging of their population. So I really believe that historical demographic data can be a powerful tool, and that demographers should be involved in their study. It's not only a matter of here is the data, do your study. I think demographers should, can, and should be involved. And I might be wrong, but I believe that in the coming years, one of the major issues will be to find a balance between research opportunity and really optimizing the potential of these data, and also all the privacy and confidentiality concerns that pertain to these data, and the fact that more and more we want to link various data sets together, and that raises issues as well. So thank you very much to you, and thank you to the Canada Foundation for Innovation for funding our project. And don't worry, it's not like that at this time of the year, but that's a picture from the Saguenay region. Thank you very much.